the issues with designing drugs is that no matter how good these drugs are with all of these tests that we do in the lab, on bench work, on cells, none of it really matters how well your drug can work fighting Alzheimer's if you can't get it to where it needs to go. In Alzheimer's, that's the brain. So some of our previous molecules, this guy here is what we call an OH-pin-3. It was a great antioxidant. It fought off these reactive oxygen species, these radicals, but it could not or could barely get through the blood-brain barrier, and so it couldn't get to the brain where Alzheimer's is actually happening. So one of the things we focused on is optimizing the pharmacology of these drugs to try and get them up to that brain to target it. We've had a few approaches to this, but one of the main ones is adding more of these lipophilic, greasy molecules to be able to cross through that oil-like blood-brain barrier from the blood and be able to pass through it and get into the brain to where they can be delivered and be more effective.
Then we start sketching out ideas, and we'll use programs to write out the reaction schemes and write on the board just like you might see in a movie. And so we use that information for reading the literature to do that. And then from there, we'll go to the lab and make what we designed.
And so that's what this golden yolk here on the boiled egg plot represents. Drugs that can be absorbed through your intestines and then travel through your blood and up through that blood-brain barrier into your brain to act where they need to fight against Alzheimer's.
That's a really difficult thing for a drug to survive, and then we've got to get it to the brain. That's a lot of where the time and energy is taken in designing the molecules, and that's where we are. That's why we're trying to make it more like oil and water, and that stage of the things is where we are and what we're really fighting to get towards.